Scene script. Have you ever wondered what makes you? The answer lies in something incredibly small, yet unimaginably complex. DNA. DNA, or deoxyribonucleic acid, is our biological instruction manual, the blueprint that guides the growth, development, functioning, and reproduction of all known living organisms and even many viruses. This blueprint is written in a language that's been perfected over billions of years of evolution, and like any language, it has its own alphabet and grammar. The alphabet of DNA consists of just 4 letters, A, T, C, and G. These represent the 4 chemical bases, adenine, thymine, cytosine, and guanine. However, these letters don't stand alone. They always pair up in a specific way, A with T and C with G. These pairings form the rungs of a ladder-like structure that twists into a shape we're all familiar with, the double helix. This double helix is a marvel of nature, a microscopic wonder that's less than 2/millionths of a millimeter wide, yet can stretch out to nearly 6 feet long in each of your cells. And within this tiny space, the 4 bases combine in countless ways to write the story of life. The way these bases pair up is no random event. It's a meticulous process that ensures the stability of the DNA molecule and the faithful transmission of genetic information from one generation to the next. And just like how a specific sequence of letters forms words and words form sentences, these base pairs come together to form genes. Each gene is a unique sequence of base pairs that provides the instructions for building a specific protein. And it's these proteins that do the heavy lifting in our bodies, from building tissues and fighting off infections to carrying oxygen in our blood and helping us think, feel, and move. So the next time you look in the mirror, remember this: you are a unique expression of a genetic code that's been billions of years in the making. Just as letters form words and sentences, these base pairs form genes, the instructions for building and maintaining your unique self. Now that you understand DNA's structure, imagine it as a language, a code written in 4 letters, A, T, G, and C. These letters represent the 4 chemical bases that make up the DNA molecule: adenine, thymine, guanine, and cytosine. Together, they form a language that is universal to all life forms on Earth. This language is read in 3-letter words known as codons, each of which corresponds to a specific amino acid. These amino acids are the building blocks of proteins, which are responsible for nearly every task in our body, from building tissues to fighting diseases. This genetic code, organized into specific sequences, forms genes. Each gene holds the instructions for creating a particular protein. But here's the fascinating part. These genes express themselves in different ways, leading to the myriad of life forms and traits we see around us. It's amazing, isn't it, how 4 simple letters can encode the incredible diversity of life on Earth. Ever wondered why you have your mother's eyes or your father's height? That's genetics at work. Let's dive into the world of inheritance patterns. You see, every human has 2 copies of each gene, one from each parent. These genes can be either dominant or recessive. Dominant genes are like the class overachiever. If you receive a dominant gene from either parent, that trait will show up in you. On the other hand, recessive genes are like the quiet students in the back of the class. They only come forward when there's no dominant gene around. So if you receive recessive genes from both your mom and dad, that trait will be present. This complex dance of dominant and recessive genes is how traits are passed down from generation to generation. It's why you might have your grandmother's curly hair or your great-grandfather's love for jazz. So, the next time someone says you look like your parents, thank your genes. But what happens when the genetic code gets mixed up? The consequences can be severe. Imagine your DNA as a recipe book. If a word is misspelled or instructions are jumbled, the dish might not turn out as expected. In the same way, mutations in the DNA sequence can lead to genetic disorders. These disorders range from mild to severe, affecting various aspects of our health. For example, cystic fibrosis, a condition affecting the lungs and digestive system, is caused by a single mutation in the CFTR gene. Huntington's disease, a neurodegenerative disorder, results from an expansion in a specific region of the HTT gene. Now you might be thinking, are we all at the mercy of our genes? Well, not exactly. Enter the concept of gene expression, where not all genes are active at all times. Factors like stress, diet, and exposure to toxins can influence which genes are turned on or off. This is known as epigenetics. So while you might have a genetic predisposition towards a certain condition, your lifestyle and environment can significantly influence whether that predisposition becomes a reality. This intricate interplay between genes and environment is a key aspect of modern genetic research. While genetics can sometimes seem like a roll of the dice, remember that it's not all in your genes. Environment plays a crucial role too. What if we could correct these genetic mistakes? The future is here with gene editing technology. Let's take a moment to marvel at the wonders of gene editing, particularly the revolutionary CRISPR-Cas9 system. This innovative tool, often likened to molecular scissors, allows us to precisely cut and paste genetic material. It's like having an editing suite for our DNA, enabling us to correct genetic errors that lead to diseases. Imagine the possibilities. We could potentially cure genetic disorders that have plagued humanity for generations. Conditions like cystic fibrosis, sickle cell anemia, or Huntington's disease could become a thing of the past. But it's not just about curing diseases. The potential of gene editing extends to enhancing our capabilities. Could we tweak our genes to become stronger, smarter, or even to live longer? These are tantalizing questions that the future of genetics might answer. However, every coin has two sides. While the potential benefits are enormous, gene editing also raises ethical and safety concerns. Who gets to decide what constitutes a better human? And how can we ensure these powerful tools won't be misused? The journey to the future of genetics is filled with promise and peril. We are treading a path that could redefine what it means to be human. As we stand on the brink of a genetic revolution, one thing is clear. Understanding our DNA is understanding ourselves. And that, my friends, is the real secret of genetics.